jumping off point. So yes, I'm calling this, hopefully Thomas can correct my pronunciation, smorgasbord of multiomics data analysis. How was that? Okay, good. Yes, I call it that because this is a Swedish class, so it's a bit of a reference there. Okay, so, and there's a picture of a delicious smorgasbord. So we're pitching this talk at people who maybe have some omics data, possibly from multiple different types of omics, like microbiome, metabolome, and genome data, or even if you just have one type of omics and you're interested in doing some analysis on that. And I am not an expert in this topic myself, so it's kind of a high-level overview. You know, hopefully you won't stump me with any questions about the nitty-gritty of it. So what will we talk about here? I'll give you some resources that you can look at, and you can access these slides to get the links to all that stuff. We're going to give a conceptual overview of some of the methods. And these are the different demos that are part of, that I got from this Swedish course. MOFA, a couple of them are based on, we might only have time to do 2 of these, so we can we'll do the first one and then we'll decide whether we want to do 2 or 3, since this is kind of designed to be 3 or 4 hours, but we only have 2 hours. The first one is, 2 of them are about multiomics factor analysis, and one of them focuses more on the kind of data cleaning and pre-processing steps that you would have to do, and one focuses more on kind of post hoc analyses you can do after doing the factor analysis. And then we're also going to do, if we have time, a partial least squares type of analysis, which I'll talk about what those are when we get to it. So let's just start by going through some of the resources that are out there that you can consult. One great one is a YouTube-based and also text-based short course that the American Statistical Association put out. I would recommend looking at that. Again, you'll have access to the slides with all these links to look at. So that's one that's out there. Mixomics is a R package that is a platform for kind of omics analysis, and they have a bunch of self-guided courses. They have some webinars about different topics. They have, actually it's going on right now, a kind of self-paced online course. Hopefully the materials from that will be available even after the course is over. Yes, Marina. I believe the course is free. I have to confirm that. I think they have a lot of free materials. Some might be for a fee, but they definitely have free training materials for sure, at least some. The one that we're basing our stuff on today is the Swedish National Bioinformatics Infrastructure course, which is great. And unfortunately, you can't actually enroll in the courses because they're only for European nationals, but they put all their materials open source, which is awesome because then people like us can benefit from them. And okay, so those are just some of what's out there. I'm happy to help you find more if you have something specific you want to look at. So let's talk about what is integrating data from multiple omics and how we might go about doing that. So what do these data look like? What are we talking about here? So we call it multi-omics if we have anything that has more than one omics data set taken from the same samples or the same individuals. Like there's a lot of, any type of genomic stuff, methylation at different sites, metabolome, transcriptome, so what's being expressed, and microbiome, of course, as well, among others. And these sometimes are organism-wide, or there's also a lot of single cell approaches that give you omics data from many cells from the same sample as well, which gets into some really big data kind of situation pretty fast. And most of these omics data come from studies where we're testing something. So we usually have metadata or covariates about the organisms or cells, whether that's an experiment, they might be from different treatments, or it's covariates, observational level, age, sex, and so forth, or what tissues the different cells come from. We're doing single cell sequencing. Oh, by the way, I originally wrote this talk again for the people that do human nutrition in Little Rock. So a lot of the examples kind of come from human studies, but hopefully it is still relevant even if folks are not working on that. So what kind of questions can we answer with this type of data? Well, we can look at relationships within the omics. How are they related to one another? And we can also ask how well each of these omics in isolation or combined together can predict the outcomes, or what is the effect of the experimental treatment on some of the microbiome or on gene expression and so forth. That's very broad. Obviously, there's a lot of questions within that. Now, we are going to kind of think broadly. This is not just for omics, but in multivariate, because we have many variables in our omics data sets. When you're doing a multivariate analysis, there are kind of two broad categories that you can divide them up into, which is supervised and unsupervised learning algorithms. What's the difference? Supervised is we have some kind of outcome variable, like in a lot of these human studies, it would be like the disease status of an individual, individuals with the disease or without. And we're trying to figure out what combination of features in our omics data sets best predict that outcome. Like, could we look at the omics and see if that patient has a high probability of getting cancer or whatever? But there's sort of like there's an outcome, not just the omics data, there's also that outcome in it. Unsupervised does not have groups that the different observations belong to that we're trying to classify. We're just exploring relationships between the data sets and kind of trying to find patterns in there without them being associated with a particular outcome. So any type of analysis we do is either going to be supervised or unsupervised. That's like the broadest category you can divide them up into. And within, now just thinking about supervised, if we have an outcome in mind, there's kind of two categories within that. There's descriptive supervised and there's predictive. Descriptive is we want to find weights, parameters basically for the different variables, the different like taxa in your microbiome, or the different sites in your methylome and everything. We want to find weights for each of those that optimally explain variation between our different classes, our different outcomes. What does that mean? Optimal, there's a lot of different ways to do that. We'll get into just some of them. And predictive is more about if we have our data set where we have a lot of omics data and we know their outcome. We want to, in a goal of a predictive analysis, is not necessarily explaining why they're different. It's trying to maximize the ability to predict the status or the outcome for new data that we do not know their variables. Like for Soheila's, if you had allergy status is your outcome, you might want to get like new samples of microarrays from different patients and predict if they're allergic without actually having to, just from their omics. And so you basically have to construct a rule using the data that you have and then apply it to the new samples that don't have a prediction in mind. And then there's it's kind of easier to tell if you have a good predictive model because you have a very natural way to say if it's good or not. Like, did it predict or not? Like, is your weather forecast good? Well, you can see if you're right or wrong by looking out the window. So like it's a predictive model is kind of easy to tell if you did good or not, because prediction accuracy rules the day. But sensitivity and specificity are kind of like the false positive and false negative rates we've been talking about. Those are kind of the metrics you use to tell if you have a good predictive model. Any questions so far? So what methods are we going to use? Well, it depends on how big your different omics data sets are. They vary in orders of magnitude of how many variables they have. Like metabolome data tends to be kind of small because it's very expensive to generate metabolome data. So usually there's only maybe a few dozen or at most a few hundred features, different metabolites in a metabolome data set. They're on the small end. Microbiome and transcriptome data tends to be a bit bigger and can have thousands. And but then when you get to genome and methylome, you have, incredibly large numbers of loci, and so you have maybe many thousands of features in those. They vary by orders of magnitude. And so we call this the N, or the P versus N of the data set. P is the number of predictors, the number of different variables. It varies a lot. Like we just showed different P from 10 all the way up to thousands. N is the number of samples. It doesn't vary quite as much because this comes from research in, well, whether it's clinical or just life sciences in general, it's individuals. It's probably a smaller number in the dozens, sometimes a little bit bigger, but it's not huge. So if you have, I was talking about this with Thomas just now, the relative size of the number of variables and the number of individuals, size of the data set determines what method you use. In situations where you don't have too many predictor variables, you can kind of go with the Bayesian approaches. In the middle is where the classical frequentist statistics lives. But then once you get to having way, way more variables than you do sample, then that's when you start to go with kind of machine learning and deep learning approaches. So it really depends on how your data is shaped. Is it long or is it wide? And there are quite a few different platforms. R has a lot of packages for this type of analysis, and Python as well. And there are also some sort of like more graphical interfaces. There's one called MetaboAnalyst that is kind of a more beginner-friendly graphical interface for looking at metabolome data that some of you may have encountered before. Today, we are going to use R, but actually, behind the scenes, some Python code is going to be run from within R, because some of these algorithms are actually written in Python. But you can run Python from within R, or vice versa. This is a famous picture of a chart of how data scientists spend their time. And you can see over half of it is cleaning and organizing the data. Only a small percentage of the time of data science is actually building models and getting cool answers. It's all about cleaning and pre-processing the data. And that's, the omics data is no exception there, because you end up churning out a lot of raw data and you need to get it into a format that can be put into these models before you actually do it. So one common step is if you have variables that are almost all zero for all individuals, they basically have no variance. All that's going to do, if you keep those in the model, is slow down your model and take it longer to get the answer. So you want to clean up the data first by getting rid of some of if we're trying to explain variation between the groups and your variable has no variance, it can't possibly explain variation between the groups. So you might as well not include it. Missing values is going to be a big issue with these data sets. If some samples have too many missing values and some individuals have too many missing values, they're also going to just add noise and not help you. So you should take steps to clean those up as well. And yes, there are imputation methods, ways to impute missing variables if you have kind of a moderate number of missing values. But if you have too many, that's going to be no imputation in the world is going to help you, because you're basically don't have data anymore. You just have kind of modeled missing values, which is, you have to have some data to start with. So you can't really, if you have too many missing values on an individual, you tend to have to exclude them from the model before you start. So if we start a omics analysis, a good place to start is to do some simple multivariate techniques like PCA, principal components analysis. It's kind of like the granddaddy of multivariate techniques, and they are pretty quick to run, and so they can be a good first step, especially to do visualization, and it helps you understand the structure of the data. So it's often a good first step, and if there's biases in the data, it'll pick it out. Like basically what a PCA does, here's your data, your N by P individuals going down and variables going across. It finds the optimal way of a few component vectors, so a few variables that can explain most of the variation in this huge matrix. Basically, the product of a loading vectors and component scores is your PCA. That's kind of what a lot of these methods are doing, is taking a matrix that's huge and reducing it down to a minimal number of components that can capture most of the variation in your data with fewer variables that you can then make conclusions about. So Haley, you have a question. So the component scores are the loading vectors. You can see this one is kind of, yeah, this one is, these vectors are n long. It is a score for each individual in the data set. So if you had 20 patients, this would be 20 long, and however many components you use, like this one shows 2. So we're reducing our data down to 2 dimensions, and the loading vector is p long. That's how many. So if you had 1,000, 1,000 variables in your omics data set, it would be 1,000 long, and that's like the loading of each of your genes or metabolites on those dimensions. It explains how much each of those raw data variables is contributing to the reduced one that we got. So that's a great point, because that's kind of fundamentally what we are trying to do in most of these methods, is reduce it down to fewer dimensions and figure out how each individual is associated with those dimensions and how each of the variables in our raw data set is associated with those dimensions. So what are some of the techniques? We're going to look today at multiomics factor analysis, and also, which is an unsupervised method. It does not use the outcome variables in it. And we're also going to look at SPLSDA, which is called sparse partial least squares discriminant analysis. We'll go over what that means in a minute, but that's a supervised algorithm. It does use the groups in the analysis. And there's a lot more methods out there, which we won't go over today. There's a bunch of more machine learning type methods that are kind of to do dimension reduction and make cool plots. Neural networks, network analysis methods. I do not profess to know much about any of them, so I won't try to teach them to you, but they're things you might be interested in looking into. But I do just show a couple slides about them just so you have an idea of what they look like. Like this is, there's a bunch of sort of machine learning ways to reduce dimensionality in the data set. I kind of think of them as PCA on steroids, and they can be used to integrate information across multiple omics data sets. Like this, basically, if you give it points with the different continents, the colors are how it assigned points to different continents without actually knowing what continent it was part of. So you can see that it does a pretty good job of pulling structure out of a data set and finding patterns. So that's like an unsupervised algorithm that actually was able to pull out what we all know is the pattern in the data set. I'm not sure how it actually told Africa apart from Eurasia, but somehow it did. So, kind of a flashy thing there. And deep learning methods, I know hardly anything about it, but they're really cool. But the performance of them scale is not very good with small data sets, with small numbers of individuals. So in some of the data that we work with, they aren't the best, they really start to shine when you have huge data sets. That's when they really outperform a lot of the other methods, but they're very data hungry. So they may not be optimal for some of the data sets that we work with. But basically, it can be boiled down to some kind of dimension reduction component where you reduce the number of variables down and then clustering on those dimensions. A good advantage of deep learning is that it's really good at picking out nonlinear patterns. It is not as constrained to linear patterns as some of these methods are. And what a lot of people do in practice is do like a deep learning on the data and then take the results of that and stick it into one of those machine learning methods I just mentioned to actually visualize the results. That's kind of what the cool kids are doing these days. I'm not one of them. And then there's a bunch of network analysis methods where you can kind of use things like nearest neighbors to find connections between individuals. And then kind of there's ways to make networks from each of your omics data, and then there's techniques that can fuse them into one consensus network. Again, it's just something to be aware of, but we won't go into it anymore today. But here's what we're actually going to do today is multiomics factor analysis. And this is a hypothesis-free, in other words, unsupervised. We are just trying to find patterns in the data without having a certain outcome in mind to explore. Sorry, this is a little small to look at, but basically the goal is to take your different omics layers. So like, let's say you have metabolome, genome, and proteome data. These are your raw data for different, well, the individuals are going across and the variables are going down. Again, it's a lot like PCA where you're turning, you have these W matrices that you pull out how each feature is associated with each of the factors. Those are like the components of a PCA times a Z matrix, which has one column for each individual. But again, so this is, but it integrates across all the omics in your data set at once. And then you can break down the variance explained by the different factors. Hopefully this will make more sense when we actually go through the demo. So let's hold off on questions yet till we actually see more details. And another cool thing about it is in the process, if you have missing data, you can actually use the results of the factor analysis to predict missing values in the data set if you have a modest number. And then kind of the goal is once you have these low dimensional factors, reducing your huge number of variables into a few, you can look at patterns in them and see if you can come up with a biological interpretation for those. And the other one we're going to do later is partial least squares discriminant analysis. If you look at the figure for this, it looks almost exactly like the figure of PCA. Again, they're all kind of different variations on a theme of taking a big matrix, reducing it into components that are associated with the different variables in your data set and loadings for the different individuals. Or the other way around, but yeah, it's a supervised method because you can see the difference with PCA is that it has this Y vector, which is like the experimental treatments or the groups. So it's finding variables that best explain variation across the individuals, but also maximally separate the groups, whether that's the treatments or the disease outcomes or whatever. And you have to kind of choose a number of groups, which is usually your, the different treatment levels. And then it's trying to maximize the variability within groups, between groups, and minimize variability within groups. Just kind of like an ANOVA, but with a lot more dimensions. And what we're going to do sparse partial least squares, which can actually sparse, meaning having a lot of zeros in it, because it's just like the PLS, except that it tries to reduce the complexity of the data even further by turning a lot of the loadings for the different variables to zero using a variable selection approach. So we can further simplify and avoid overfitting the data. And you use cross validation to determine that, which we'll talk about when we get to it. And again, since it's more of a predictive model, you get a new data set to predict on or use cross-validation within your data set to assess the performance. We'll talk about what that is when we get to it. Yeah, so let's get it started and let's analyze some multi-omics data. So that concludes the introductory material. Let's take a second for questions, if there are any, before we go on. Any questions? Cool. So hopefully I haven't blown your minds too much yet. What we're going to do now. So here, if you go back to the agenda page, we just went through these intro slides. I've got basically we got these different demos, and I have them. They're like code notebooks. You can use the HTML version to look at the text of it. But what we're going to do is we're going to go to the Posit Cloud demo server again, and then we're just going to run. We're kind of going to walk through a multiomics factor analysis step by step. It is not crucial that you run this along with me because you don't really have to type anything. You're just going to like kind of click run whenever you get to some code and like see the same thing pop up on your screen that I have. I wanted to make that option available to you if you want. Do not feel obligated. You can just watch what I do and be wowed by the pretty colors flashing on the screen and stuff like that. But yeah, it's up to you if you want to actually run it yourself. You have that ability because it's all installed and ready to go. So let me log into that and I'll get mine started. Now everybody's is in here. Let's see, here we go. Yes. Yes. They should be right here that we just looked at. Yep. So if you click on that link, you should get to them. It takes a minute to load. Yeah, it's kind of a big file. Yes, it should be there. I can email them later on if you're having trouble getting to them from the website. It could just be that they're slow to load, but they should be there. Because I just pulled them up from the website too. I didn't pull them up off my computer. I just went to the website as well. So it should work for you as well. Okay, let me just clear everything. Restart R from yesterday. Clear the workspace so that we can start from a clean slate. Go to the root project directory, open up the day 2 folder, and go to the demo1.rmd file. And yeah, this is basically the raw data for this notebook. It's not just the R code, it also has some text explanations interspersed with it. But then anytime you see a little block of code that says R on it, you can run that code by just clicking this green button and it will run the code in the console. You really shouldn't ever have to modify anything. As long as you run each block in order and don't skip one, everything should work exactly as I'm doing it. So what we're going to be looking at is this demo. In this demo, there's a single cell sequencing data set with 3 different omics from mouse cells of 2 different types. There are 2 different sort of cell lines. Some are stem cells and some are embryonic bodies. Don't ask me what that is, but that's what we're looking at. And in each one of those, any cells from 2 different lines or 2 different types of tissue, basically, and in each cell from each type of tissue, they did 3 types of sequencing on those cells. So each cell was sequenced 3 different ways. Chromatin accessibility, methylation, and gene expression, RNA-seq, was done on those cells. So we got 3 omics from each cell, lots of cells from each individual, 2 different tissue types within each individual. This was a paper, it's all from a publication, and there's the link to the paper. Now we are going to run this code block. We're going to use the Mixomics package as well as the MOFA2 package. And you can see we're starting a virtual environment. And what that is doing is basically the MOFA algorithm is actually in a Python package. I set this up so that Python is installed on this demo server and R will talk to Python, do the pre-processing in R, pass that to Python. It'll actually do the factor analysis, bring the results back into R for plotting. You can set this up on your own machine as well. It takes time and troubleshooting to do. If you really want to do this on your own machine later, I can help you. But for now, just take my word for it that it works, and we can deal with cross that bridge when we come to it. Yes. Yes, and all of it can be done on SciNet as well. I would say it's easy, for me at least, easier to do on series because R and Python are already installed there. So configuring the virtual environment is quite easy. You can do it with like basically 1 line of code, and that's basically a Python version that's set up with all the Python packages that you need. Basically, there's just 1 Python package that's being used here. So yeah, you can do it on SciNet, and if that's what you're into, I highly recommend it. And you can see I've also in the data folder, I have the data for today, the RNA-seq and the different methylome and everything all in there. So what we're going to do is we're going to just explore the data first before we actually start doing complicated models on it, because it's not very smart to do complicated models on something you don't know what it is. So if you run this, what we're doing here, basically reading the data, reading a lookup table that turns the sort of numerical identifiers for the different variables in the RNA-seq data set into more descriptive names of the genes that they're transcribing, and then match that raw data with the lookup table so we have more descriptive names of the variables. That's all that's doing. And then printing row 1 through 5 and column 1 through 5 of the RNA-seq data set, as we learned yesterday how to index data frames and print some subsets of them. So we run that, and then the results will pop up underneath the chunk of code. Or well, actually, no, it's set to pop up in the console. So that's good. So basically it's a bunch of continuous variables. The transcripts have a continuous measure of abundance of them that can be 0 or however high of a positive number, right? So here are the columns are the cells, so ESC, embryonic stem cell, and then the rows are the different transcripts which have names and their abundances. So far, so good? Okay. What's next? Well, we do the same with this bisulfite sequencing. That's the chromatin accessibility and also the methylome sequencing. Yes, Sohaila. Yes, so I uploaded the data onto this demo server. If you look at this data folder, this has all the data from a couple days, but there's basically, the data are just text files. Here, I'll just open one. Hopefully it doesn't take too long to open. Yeah, we'll open it anyways. Yeah, right. It is a text file, and so as long as it is in the proper format to be read in by this function, read delim, which reads tab separated text files in as data frame, as long as it's in that format, yeah, but again, whatever comes out of your sequencer may not be in that format. So there's probably other parsing steps that are done before this, which, yes, that is a separate question.
So Haley, I think you're also asking, like, It can be on your computer, and that part
Yes.
right after read the lim where it says data slash side dot text, that's the path to where that file
That's
is.
right.
Like you might be more familiar with C:// plot plot plot.
That's right. That's right.
So it would look like that wherever.
Yes.
There's the shortcuts to like copy and paste like path or something, but
Yes.
that's what it needs.
There is a
where the file is.
this only works with data slash because I put a folder named data into the directory that R is running out of. If you want to get it from anywhere on your machine, you would have to put the full path in like C/users/my name/documents. Thanks for saying that. Yes. So that's if you're having a problem with that, I can help you for sure. Yeah, so then I've loaded the other two, and you can see these other two data sets range from 0 to 100. They're kind of like percent methylation and percent chromatin accessibility for each of the loci, basically, of each individual. So they are not from 0 to, however, they're only from 0 to 100, right? So they're different. The three omics have different structure to the data. But they all come from the same cells. Like ESC_A02 is the first column of this data set. And it should be the first column of all the data sets. They're already sorted by individual in the same way of all three. So that's another thing. The naming of the individuals must be consistent across the omics. Otherwise, how's it going to know which is which? Yes, Thomas. No, it is not missing. That means 0 methylation was detected versus 100%. Yep. Missing would be Na. Yes. I forget if we have that issue coming up, but we'll see when we get to it. Okay, so here's kind of some data pre-processing that you might want to do if you have this kind of omics data. So we're starting with the RNA-seq layer. And that's the first one, which goes from 0 to however much transcript abundance expression there was for each feature. So, and you can see a lot of them have high numbers, but some have zeros, and some of them actually have zeros for most of the individuals. So, what's a good way to deal with that is to only do this analysis on highly expressed genes. And so basically, I don't know why this number was picked, but the analyst decided that having a mean of 1 across rows or greater is a high enough level of expression that we don't think it's just noise. So, and then also do a log transformation to kind of normalize the data a little bit. And again, because we have, so basically we're going to subset by where the row means are greater than or equal to 1. This means keep only the rows greater than or equal to 1, but all columns. And then take the log of this plus 1, which is the offset because we got zeros in the data. You can't take the log of 0. And then we're just going to print the first few rows and columns again to see what the new data looks like. And then use the dim command, that means dimensions, which will say how many rows and columns our newly reduced data set has. We'll do that for RNA-seq. So you see it's now instead of going up to 500 and whatnot, it goes only up to 2.7 because we did log base 10. So you know it's somewhere between 100 and 1,000, and we still have some zeros, which is the log of 1. And then we end up with 12,000 rows. So those are the different gene transcripts, and 113 columns. That's how many individuals, or how many, it's actually how many cells, because the samples are cells, not individuals. 100-ish cells, 12,000 genes. Okay. Now, the methylation and chromatin accessibility that range between 0 and 100, it turns out there's mostly either zeros and 100s and not very much in between. So for a sort of simplification step, what what we're going to do is first use this function called near zero var, which will get rid of features that have almost no variance in them. So it'll pull out those. And then basically get rid of the variables that don't have very much variance of both the bisulfite chromatin accessibility omics data and the methylation one. We're going to do that for both of them. So let's run that. It takes a second. So now, yes, so now we end up with, it got rid of a few thousand variables in the bisulfite sequencing data, and we end up with about 5,000 variables in this, again, the same 113 cells. So it's good they have the same number of columns, but different rows. And then the other methylation one, methylome, you have about 11,000 variables, same number of cells. Everyone kind of following what's going on? Hopefully, conceptually, we can follow even if you don't know what every bit of code is doing. But so far, this is all fairly straightforward in terms of the code. We aren't doing anything super sophisticated yet. So let's look at the histograms of the 3 omics data to see what kind of the data look like and how they should go into the model. So we're making a histogram of the RNA-seq, bisulfite, and methylome data, just taking all the numbers and all the individuals put together and making histograms. A simple step for data analysis. So here's we'll start with the RNA-seq. You can see, right, because we did a log transformation on data that went from 0 to a high number, is kind of skewed-ish, but it's still, basically, this is considered to be close enough to normal that we can model it as a Gaussian or normal variable. It's close enough, because things don't have to be perfect. They just have to be close enough. But what about this bisulfite? Well, obviously, that's not remotely close to normal because it's got, it only goes from 0 to 100, and it's not bell curve, it's the opposite. It's got like high and low and nothing in between. It looks more like a binomial or binary variable that's just kind of spread out a little bit on those 2 peaks. And methylome, similar. We have like some 100s and then a bunch of low ones. It's kind of got 2 humps as well, nothing in the middle. Right. So, yes.
That's sort of like what I was asking before. Like, to everybody's eyeball, there's clear ways to decide how to in
right.
This kind of a thing.
Right. Yep.
Is there a statistic?
And that is exactly what we're going to do, is use the eyeball test here and divide it at 50, because there's not very much at 50. So 50%, we're just not going to do a formal test. We're going to.
But I can't do that 100,000 times. But I need to.
Well, this is a histogram of the entire data set. We're splitting at 50 for all.
But I'm looking at a different data set that has that change, where it's not.
Right. Yes, you can. You can do a one-dimensional cluster analysis on 100,000 times, and then that would determine the optimal cut point. And there's special algorithms that do it really fast, so you can do it on like a lot in a row really fast. That's what you do. But here we're not going to mess with that. We're just going to. You could probably do that for every locus separately here and find some optimal cut point, but we're not going to do that. We're just going to, for simplicity's sake, split it at 50. So basically, that's what we're going to do. If else less than 50, so basically anything less than 50 turns to a 0, anything 50 or more turns to a 1. We're doing that for both the bisulfite and the methylome. And then we're going to do that variance pruning step again to remove low variance features, because now we've reduced it to only 0s and 1s. There now might be some that are all 0 or all 1. We can get rid of those because they're not giving us any information. So we run that. And now we've gotten rid of some. The bisulfite is down to 4,500 variables now, and the methylome is down to 9,400, where before it was like 11,000. So we've gotten rid of, 10% or so. So that was our data cleaning. Now we can run the multiomics factor analysis. So how do we do that? Well, basically, you have to format these omics data sets into the format that can be processed by the basically the Python function that runs this algorithm. And there's sort of a special function in the MOFA package that lets you take these this list of matrices and make a special object that goes into it, and it has special methods that allow you to plot the data set or overview, make summary statistics. So let's do that. So this shows in this particular example there are no missing values. If there were, there would be gray blocks for the individuals with missing values. This one doesn't have it. The other demo does have some missing values. This one does not. Now we have to set up the model's tuning parameters. And this is kind of something that probably has default values, but you kind of have to have some experience with a method to know what the appropriate tuning parameters are. But first of all, we're setting Gaussian, Bernoulli, and Bernoulli on the response distributions, again, because our, what was it, the RNA-seq data kind of was more continuous, and the other two are binary. Bernoulli distribution means zeros and ones, so we're going to set those, name them so that we can have figure labels that are named. Yes, question? Yes. N is the number of, yes, individual cells that were sequenced. Yep. Yeah, that was called P in my slides, but the number of variables. So like in the RNA-seq, we have 12,000 genes that we're looking at the expression of. And then in the methylome, there are like 9,000 sites on the genome that we looked at methylation at those sites. After we got rid of some variables that didn't have variants in them. Yes. Well, the variables are unique to each data set, but the one that's common across them is the individuals. Every cell has an ID, like cell number 1 is going to be the same cell number 1 in the 3 data sets, and it has to be for us to integrate across the omics. Yeah, you'd have to go look at the paper. I'm not 100% sure whether they come from actual different mice or if it's all from the same mouse in different cells. I'm not sure, but basically, I think they might even come from the same individual mouse. They are 113 different cells from they took some cells from the stem cells and some from floating embryonic bodies. So there's kind of 2 groups of cells that we're looking at how the gene expression differs among them. That's basically what we're doing. Yep. Well, all 113 cells were sequenced for the same 12,000 genes in the RNA-seq or transcripts. So okay, I see. So yes, that's a methodological question that I don't know about, but like I guess if you do single cell sequencing, there has to be a barcoding step to track what cell the different transcripts come from, so then you can keep that identifier after you pull out get the data from the RNA-seq. I don't know. I'm only a statistician. I don't know about that. So yeah. Okay. Right. In this case, there's no missing ones, so all 113 cells have all 3 omics. You can still do it if some are missing some. This one does not. Yes, so basically we have to, and the other kind of the one option that maybe is a little bit more obscure of how they did this was we're going to do 20 factors in our multiomics integration, right? We have 12,000 RNA-seq, 4,000 bisulfite, and 9,400 methylome. We want to reduce that into 20 variables to explain the maximal variation. Why is it 20? I'm not sure, but that was determined to be a reasonable number to reduce it to. Too many would be hard to interpret. Too few, you're going to lose information. So somehow 20 was chosen ahead of time. And out of those 20, it was decided to automatically drop factors that explain less than 3% of variation across all the omics. That's the threshold for retaining the factors in the final model was set to 3%. Again, I am not really sure how they arrived at that number, but it was found to be kind of a sweet spot, I guess. Maybe it was tried with different tuning. Again, you probably would want to do, if you're really doing this, you could probably do a cross-validation to determine the optimal threshold, because we're not going to get into that in this demo. We're just picking one. But probably, I would guess, if you were really doing this, you would want to run the MOFA many times with different thresholds, look at the out-of-sample prediction performance every time, and pick the one that optimized it. But we're just gonna pick these. We're just gonna assume them for this. And now we're going to run it. And so this run MOFA function is going to connect to a Python package externally, and again, this takes a couple of minutes to run, because it goes through different iterations running until we reach the optimal solution. And while that's running, and so every iteration is printing some output. But this is actually running, Python is actually running this behind the scenes. Yeah, so basically there are some plots we're going to look at once this is done running. And we're going to look at the variance explained by each of the factors that were identified in the final model as explaining the most variation across the omics. What % of variation in the data do they explain? We can look at that as tables and as plots. Like, is the RNA-seq most explaining the variation, or is it another one of the omics data sets? So that's the first thing we're going to look at once this is done running. It should be done in a minute or two. Any questions while we're waiting? Yes, Greg.
Could you use like 2 time points as like the 2 omics? Just kind of look for like an interaction. Like say you had RNA-seq from.
Yes, I think you could do that. Yes, that would probably be. Yes, you're integrating information from multiple omics. There's no reason why it has to be qualitatively different ones. I think you certainly could do it for different time points, and that would be a great idea, I think. Yeah, and it should, because it's sort of tracking individuals across them, it would account for the fact that they come from the same individual. Yeah, I mean, I think just off the top of my head, I don't see why not, and it seems like that would be a good idea. Yes. Right. I had to configure that ahead of time. I had to install Python on this demo server and set up a virtual environment. You can do that. It can be done. Again, you would need your admin to install Python for you, but you can do it pretty easily. Well, probably the best way is to do it on Skynet, because everything is already kind of installed there. And, you know, computationally, some of these things might take a long time to run. So this one isn't too bad, but yeah, I would say it's a good idea to try to do it on Skynet. Yeah, I think there's no reason why you can't set this up on your own machine. I mean, not everybody has to run it. As long as somebody in your group can run it, you know, you're good. Yeah, I think it does about 50, so we should be,
Who's gonna need to have a Python expert?
yeah, so, you know, it's,
Maybe just one for building.
that's right, yes. I don't really know Python that well, but I have occasionally used some R code that runs Python, but I'm not a very good Python coder myself. Yes, that's right, yes. Thomas is like paying close attention to the output of our iterations, and every iteration you can see number of factors is 3. So yes, it actually has winnowed it down from, so I guess 20 was like a reasonable starting point. It would have been fine to start with less than 20 because it pulled it down to 3. Using that 3% threshold of variance explained, it found only 3 dimensions that explain the maximum amount of variation in the data set. So now it's done running, and it gave some warnings, but hopefully we can ignore them. And yes, so we're down to 3 factors. So let's look at the variance explained by each factor by running this next code block. Let's do another couple of minutes, and then we'll take a break, I think, and we can finish later. But so it gives you R squared. First of all, it gives you the R squared for each of the 3 omics overall. The RNA-seq has 13%. The other 2 are around 5%. So that one is explaining more of the variation. And then you can also look at that broken down by the 3 factors that were identified as being the ones that explain the most variation. So you can see how it's broken down. And this graph actually shows you something interesting, which is that it looks like, so you can see the color of this is the percent variation explained. It looks like there's one variable, basically one dimension in the data that explains a reasonable amount of variation across all 3 of the omics. Basically, integrating the omics together, one variable is explaining a lot of the variation combined, but the next 2 factors have almost 0% variation explained for 2 of the 3 omics, and still a decent amount for the RNA-seq. So it looks like you can actually knock out almost all the meaningful variation in the methylome and the chromatin accessibility omics in one variable. Yes, and so yes, we found these three underlying variables, and the RNA-seq contributes to all of them, and some do not. So let's do a little bit of trying to look. It's like, well, what does that mean? What are these variables? They're just kind of parameters in a model. They don't mean much. Do they have a biological interpretation? One way we can try to get at that is to look at the features, the variables from the original data that are associated with the latent variables. So what you can do is, for each of the omics, plot the top 10 variables that are associated with each of those, with each of them. So we'll do that. And it makes like it's doing a couple of graphs, so we'll scroll back and look at them all one at a time. What are they? Let's see. It's doing some for each of the 3 omics. So basically, if there is a feature that that from each omic, from RNA-seq, because factor 1 was associated with variation in all 3 of the omics data sets, we might be able to find common features across those 3 omics that are related to one another. Maybe if they're related to that feature in our model, they also have some actual relationship in real life, that they're part of the same biological process that differs among these different cell lines. So let's see. So this is in the RNA-seq. You can see that there are some of the these are the names of the transcripts that are highly positively associated with factor 1, and some are negatively associated with it. And this is the same basic thing, except it just shows the top 10 whether top 10 in absolute value, and it tells you whether they're positively or negatively associated with the factor. And you can also make a heat map of the top 10. And what this heat map shows, this is interesting because it actually shows the individuals. So what we've done for the RNA-seq, we found the top 10 transcripts, the top 10 genes that are associated with variation in that. And then for each individual, we look at how much abundance that transcript had. You can kind of see there's two groups, right? Like some individuals have high expression for this group of genes and low in those. Some individual, or not individual, some cells. And some cells have high expression in this group and low in that. And it turns out, yes. Yes, so they don't really have any intrinsic meaning. They are essentially a vector of numbers that is associated with each that each of the variables in the raw omics data has a weight on those factors that will, and if you multiply those the weights for each factor on the variables times the loadings of each individual cell on the factors, that in that very small dimensional data set maximizes the variation in the full data set. So like, right, we had 113 cells, and across all the omics, we have 12,000, around 20,000 variables, right? So we sort of have like 20,000 by 100. We've reduced it down to 3 factors across all the variables. So 3 by 20,000 and 3 by 100. So we've reduced this 20,000 by 100 to 20,000 by 3 and 3 by 100, a much, much reduced data set compared to the huge original data set we have, and still explained something like 15 or so % of the variation in that very, very big data set with fewer factors. So those may be the ones that are most important for differentiating our cells from one another. Yes, it is. Yes, they are more or less in spirit the same as components of a PCA. And if you look at the heat map, we'll zoom in on it. You can see the cells. Well, it's kind of hard to see, but there's some of the cells start with ESC and some start with EB. So that was the embryonic stem cells and embryonic bodies. And if you look at it, you can see the ESCs are all the ones that are highly expressing this group of genes, and the EBs are low in that and high in the other. So it seems like even though the model did not know anything about the groups, it's actually been able to find the ones that are explaining the most variation are actually associated with those groups. So it seems like the groups truly are different in some meaningful way in terms of gene expression. I mean, not surprising because they're 2 different kinds of cells, so it would be weird if they weren't, right? But they are, and that's nice. So yeah, I think we're actually very close to the end of this one, but I think now would be a great time to take a 15-minute break and stretch a bit and kind of ruminate over things. So let's just start again at 11:35, and then we can do a couple more minutes on this, and then maybe we'll skip number 2 and look at the partial least squares things a little, because I think that might be something y'all would find interesting. So let's do that when we get back at 11:35. Thanks.
Ways backwards.
We'll get started again in just a moment. I think there's still some people trickling in. Do we have? Yes, I see. The heat map has a tree drawn on each one, and I believe that is some kind of you can, after we get the factors, it calculates a tree that optimally separates the samples. And so we can see that actually you see how there's like one main split, and that actually corresponds with the two main groups of cells that were in this data set. So I think it's just a default thing that is plotted. I'm not sure how you would go about getting the information for that tree out, but yeah, so yeah, I mean, SAS has lots of, sorry, can you say the name of it again? Yeah. Yeah, based on the pairwise correlations of the individuals, yeah, probably is. Yeah, there are many ways to generate a tree like that. So let's go ahead and finish up with this one. Actually, there's only about one more thing to show in this demo, and then we'll jump ahead and do some of demo 3 before lunch, just so you can get a idea of another method that's out there. So when we took a break a minute ago, we had just made these heat maps. Oh yeah, so this was the RNA-seq one, and I didn't show the other ones. This is the let's see, which one was this? This one is the from the bisulfite sequencing, basically accessibility of the DNA at different sites, and you can see there's like a bunch of the sites. Who knows what those different ones are, but some are positively associated with factor 1, and some are negative. And so yeah, this just shows that you can generate these plots for each of the omics separately. And once again, so here are the values for the heat map for this bisulfite sequencing. And if you remember, we transformed it so that it only had zeros and ones in it. So the data are zeros and ones. so there's only two different colors in the heat map, where the RNA-seq one had a continuous variable, so it had a lot of different colors, but it looks like one means highly accessible and zero is, or sorry, I think, yeah, one is very accessible and zero is not. So there's a bunch of sites that are associated with these embryonic stem cells and some of the embryonic bodies. And if you look at it, there's actually, yeah, maybe like these sites are associated with those same genes that were highly expressed in the different cell types. You would have to actually go in and look at that. And finally, whoops, the methylome, out of the top 10, they're all positively associated with the factor one. A bunch of sites there. And again, it looks like there's a reasonably good separation between the two groups in our data, even though the model did not know about those groups. It looks like we're separating them to some extent. Thomas. Yes. I'm not sure how the sorting of this works, but yeah, you can see that the way that the nomenclature of the data set worked, any cell that was from the EB group had EB at the beginning of it, and anything from the embryonic stem cell group said ESC. And with some exceptions, many of the cells in the EB group have all or mostly low methylation for these top 10, and then the ESC has mostly high methylation, but some are not. Oh yeah, well this ESC, I think it's they're kind of in between. I'm not sure why it's sorted in that way. Yeah. Yes, but those ones, they might be sort of anomalous. They do have a lot of the 10 that have, you know, a lot of the EB cells have zero methylation on all these top 10 ones, and those two have are in between. So it seems like the methylation is not perfectly separating those out, because it didn't explain as much variation. So it might not be as distinctive between the methylation pattern, may not be as different between those two cell groups as the RNA-seq expression profiles. Who knows? This is just kind of like initial exploration, so it's a jumping off point. And then, as I told you a minute ago, that there are, actually this might be something about it. There's that a lot of people take the output of some of these dimension reduction algorithms and put it into another machine learning dimension reduction thing just for plotting purposes. So that's what the person that wrote this demo is doing. It's gonna take the factors from the factor analysis and put it through yet another analysis real quick, a machine learning, just so we can help visualize the cells in the different groups. And I believe there are, yes, so it turns out that some of the cells are actually controls. I don't know the experimental design, but it turns out that some of the cells from each group are controls. So there's like a third group in it. So maybe that's why some of them from this heat map look different. I'm not sure. So I won't go through the code for this too much, but we're running two different visualization algorithms, and you can see here there's two different visualization plots for two different machine learning algorithms we use to visualize the separation of the groups. And you can see that this is it's all using the same visualization technique, UMAP, but it's the three different factors. And so this one, factor one is separating out them pretty well, except for one of the control individuals is sort of being placed in the wrong group, but for the most part, it's separating them. Factor two is kind of the same. Wait, let me see, why are there only two? There should be three. Oh, I see. No, this is actually the, I'm sorry, I got confused. These are the two dimension reduction plots overall, and then we can look at it colored by the actual groups that the cells belong to. And now let's color it by the factors, not the groups. Yes, that's what we're doing here. So if you look at factor one, it looks like factor one is doing a pretty good job of separating out the clusters. Like this, the cluster from the embryonic stem cells are very high in factor one, and the control group is very low in that factor. Factor two is separating the controls from the other two, but it's not really separating these groups. They both have low values for it. And finally, factor three is not associated as much with the difference between the two groups, embryonic stem cell and embryonic bodies. So those are just some other plots you can make, and that's the end of demo one. So while maybe you're left with more questions and answers about what exactly this is, it is an introduction to one way of looking at this multivariate data, especially if you have more than one omics. And actually, so let's skip demo two, because demo two is actually another one that also uses that same method, but is kind of focusing on more downstream things you can do with it. But I think it's a better use of our time to look at a different method that is a supervised algorithm, just so you can have an example of both. And this is also this partial least squares discriminant analysis. We're only going to be looking at one omic, so it kind of is a little bit simpler in terms of not trying to think and wrap your head around integrating multiple omics. It's trying to look at one omics data set and how it associates with a disease covariate. That's what we're going to be doing now for the next 45 minutes, and then quit at 12:30. Sound good? Okay, so I'll close this one, and I'm going to go ahead and clear the workspace and restart R, which will no packages are now loaded anymore. So I'm making sure I'm starting at a clean slate. And we'll open up the PLS-DA case study. And this is going to use the Mixomics package to look at, I forget what type of omics it is. It's a gene expression omics data set on 63 individuals. Yes. Yes, so first of all, I just closed whatever scripts I had open, although you don't have to. Then I press this little broom button, which will just press X on it, just close it. Yep, just press X to close it, but you don't have to. I just did to get it out of the way. The main thing is to press this, clear objects from the workspace. It's a little broom, and then go to session, restart R, which will get rid of any packages I had loaded, just to make sure we're starting from the same place. That will ensure that anything you run will do the same thing as me. So we've got 63 individuals that had they took tumor cells from individuals that had 4 different types of tumor, and want to look at whether gene expression is different across these 4 different types of tumor from people with different diseases, basically. So yeah, so there's 63 individuals, and it's not a balanced data set fully. There's 8 with the first kind, and then up to 23 for other classes of tumor that people had. So this is human subjects, and there's a little over 2,000 genes in this omics data set. So we have 63 by 2300 matrix of gene expression data. But we also have a Y, that's like our X. Our Y is what kind of cancer did they have. So that's just one value for each of the 63 individuals, what class they belong to. And then there's also, and this is built-in data that came preloaded with the Mixomics packet as a demo data. So we're not actually loading it from an external place. It's just it comes preloaded. Yes. Demo3.rmd. Yep. Yep, this is the name of it up here. And let's see. So basically, what we're going to do is a good way to start is to do PCA as kind of a first step because it's quick to run and kind of gives you an idea of the data. And then, which is unsupervised, it does not include the outcomes in it. And then we're going to do our supervised partial least squares discriminant analysis to find dimensions of the data that not only explain variation across the genes, but also variation in the outcome, the classes, what type of cancer people have. Can we look at gene expression to predict what class of tumor that expression profile came from? So select the genes that can help predict the class, the tumor type of the samples, the individuals. So again, there's like some little homeworky questions here and there in this, but we're going to kind of skip over those. But I encourage you to go back and look at those because there's answers in there as well. The questions are more about how to interpret the data. We'll go over it as much as we can in the time we have. So right, we're going to load the Multiomics package, and because we're using demo data, it just loads this way. Small round blue tumors, I think is what it stands for, and so it's just going to load, and then we're going to do a PCA on it on our 63 by 2300 matrix using the R function PCA with 10 principal components, center and scale, which means to do basically a Z transformation, a standardization to normalize or to standardize the variable each gene's expression so that they're all on the same scale. So let's do that. And here's what we get. We have proportion, explain variation of the principal components. The first one explains 10%, then or 11%, then 10%-ish, then 6, 5, 4, going down, and that plot shows that. So yeah, so then if you do that cumulative, it takes 5 components to explain about 38% of the variation in the data set. You get to a little over half by the 10th component. So it's asking how many principal components would you choose? Does anybody have an idea of what they would choose? Some people say 2 because it's kind of easy to plot 2, and also the first 2 are quite a bit higher than, they're like 10 or 11, and then you go down to a bunch that are, there's kind of a cliff you drop off after 2. So yes, 2 kind of, there's no right answer, but 2 would kind of be, at first glance of it, a good number to choose. So yes, there's some different questions. I believe this is this. Oh, so here the Multiomics package has special functions that allow you to plot some outputs of a PCA. So we're going to plot the first 2 components of the PCA, and we're going to color in the points by what tumor type it is. Even though the PCA did not use any information about the tumor type in it, let's just see if, using this simple unsupervised method, if we can pull out low dimensional information that will explain the tumor type pretty well. Let's take a look at how well it does with just this very simple method. Does it do pretty good? Not really, right? We didn't, in our naive method, get very far with just the PCA in terms of a model that can predict the tumor type. You know, if you knew the first 2 PCs of an individual, you would not be able to say which of these 4 tumor types it has. They're all over the place, right? That's not enough information. This, you can do a 3D plot, but we'll skip that. There's also this correlation plot, like a donut plot. And so for each gene, so each of those dots is one of our 2,000 genes. It shows how highly each gene is correlated with each of the 2 first PCs. So many genes have no relationship. Like if you're close to the origin, 0, 0, you're not very correlated with either of the top two. And the ones that are highly associated with the genes that are the genes that are highly associated with the PCs that are explaining 11 or so % of the variation each are the ones out here. So it's like, it looks like, a shotgun type of thing. You have a lot that are very low and a few high. So let's go ahead and do a supervised learning algorithm, partial least squares discriminant analysis, to find if we can find low dimensional representation of the data that can predict tumor class. So SRBCT gene is the if you look at, by the way, if you look in this variable environment tab, you see how there's a little blue arrow here? You can click on it to look at the individual elements that this data object is made of. So here it has gene, which is our 63 by 2000, and class is a factor, right? We learned about factors yesterday. It has 63 individuals and 4 levels. There's 4 tumor classes. And then our gene name is like a lookup table that says actually what are the names of the genes that we looked at the expression of. So basically we're making our X and Y objects from this demo data. And then if you do summary on this vector, it gives you the table of counts for each of the 4 groups, which is exactly what we thought it would be. And you can see our dimensions of X, correct, 63 rows, and our Y also has 63. So they're already been sorted so that the rows of X match up with the Ys, but that's only because it's already been done. You would have to make sure of that in your own data. So let's do the partial least squares discriminant analysis on this tumor data set using the PLSDA function with 3 components. Run that. It's actually very quick. You see it ran in a second, so that's good. And let's plot. Now, remember how we plotted the individuals from our PCA first 2 components and colored them in by what group they came from and showed it didn't have any pattern. What if we did it for our PLSDA? So now, hey, that's pretty cool. We found two variables that, first of all, out of all the genes, they explain about 10 and 6% of the variation each, but they're clearly explaining a lot of the variation in the tumor classes as well, because, well, we gave the model that information. It actually knew what the tumor classes of the individuals are, and so when it was choosing the components, it took that into account. So it's good that we were able to tease them apart, right? I mean, now if you know these 2 components, you would be able to pretty much 100% predict the tumor class of the individual because there's no overlap whatsoever, right? Yes.
So when it was unsupervised, it still had that information available too.
No, the PCA did not use the tumor class information at all. We just used that in the plot after to see how the PCA did. That's the difference of supervised and unsupervised. Supervised means we're supplying those outcomes or covariates in addition to the multivariate data that we're looking at. So it's like, well, that's cool, but how does that help us, right? Because we've given it, of course, we can get the information back that we gave it. That's not that great, but what the goal should be is to find a model that can predict new samples that we have to keep some samples back that we do not tell the model what class they belong to and see how well it can predict them. That's what we want, because then if it works on there, we could we could measure gene expression in individuals and predict their likely tumor class without knowing it. And so the way that is done, since we don't have, what we have to do is basically a method called cross-validation. So who in here has heard of cross-validation before? Well, I've said it like an hour ago, so you at least heard that. But besides that, cross-validation is a very, very, very important thing that kind of everybody should do, but not everyone actually does. It's what you do in cross-validation is, our goal is to not overfit to our data. We want to make a statistical model that captures as much variation in our data as it can, but also is not overfit to the data. If we apply that model to new samples that were not used to fit the model, we want to be able to predict them too. We don't want to draw a regression line that goes through every point in our data set. A simpler straight line, even if it has error, is better than a squiggly line that goes through every point, because that's overfit to the data and would not make good predictions about new data. We want to make predictions about new data. Because we don't really have new data, what you do with cross-validation is take a certain proportion of the data, hold it out, fit a model with the rest of the data, and then predict the ones that we purposefully didn't feed to the model. Then take another subset, hold it back, fit the model to the rest of the data and predict, and cycle through until you've used all the data points as test data at least one time. So a lot of times people do either 5-fold or 10-fold cross-validation, which means if it's 5-fold, you would take 1/5 of the data, fit the model to the other 4/5, 80% of the data, and predict that remaining 20%, and do that again for another 20% of the data, different data points, and predict, and keep doing that until you fit the model 5 times and predicted everything once. So now it's going to be, yes, it's great if you can fit a model to the data, but you want to fit one that's flexible enough to predict new data. And that's what cross-validation finds the parameters that will optimally predict new data. Very, very important thing to do. So now we actually, this will do our sparse. So we just did PLSDA, which doesn't set any of the loadings to 0. We're going to do sparse PLS, which will actually make the model simpler and more flexible to fit new data. We're gonna show it first, picking arbitrarily a number of variables to keep for each component. Then in a minute, we're going to do what we really should do, which is cross-validation to find the optimal model. But we're just going to do it first with these arbitrarily chosen numbers of loadings to keep as non-zero for each of our 3 components. And then we're going to do cross-validation to find an optimal number. So we're going to do that. So this is sparse PLSDA. So we run that. And now we'll plot our the same thing we plotted for the just regular PLSDA, except with sparse PLSDA. So you can see what is the difference between the plot we just made and the one we had before. We'll go back to the PLS. This is the PLSDA plot. And now here's the sparse PLSDA plot. Right, so the PLSDA, when we didn't do any sort of reduction of the number of factors, X variables loaded onto each factor, was able to distinguish and separate all 4 of the groups. Sparse PLS only separates 3 of them, and the green and gray ones is not have enough information to separate them. Oh, yeah, that's odd. That's true. Yes, it seems like that is a bug in this package. So yeah, that'd be something to file as a bug report. Which one is it, the shapes or the colors? That's right. I'm not sure, but very good to notice that.
Was it randomly picking 50?
It is
We're taking the
picking the ones that maximize the variation in the X and Y. Yes, it's not random. It is finding the ones that if we took if we took 50 out of the 2000, which ones optimize the A, the variation explained across all of them, and B, the separation of the groups. So then we'll plot. So you can see, remember how our correlation plot last time had like a bunch of points right near the middle that were not correlated with the components? So this is what the 50 is keeping. It's keeping ones that are highly correlated with one or more of the components, of the factors. That's why it just cut out. Now it's like a donut. It took out the ones that aren't doing very much explanation and kept the ones that are good. So that's why we use the sparse PLSDA. And here's this also shows like which the loading plot show for any of the components. It can show which genes are most associated with particular tumor classes, either positive or negative differences in their expression. So like gene 1389 is very, very low expression in EWS tumors, and then like gene 10 whatever is highly positively associated with NB. I think this is actually showing more. Hopefully, if we zoom in, it'll show more of the labels. Yeah, that's the number of bars here is not the same as the number of legends, so that seems like it's excluding some of them. Hopefully, if you wrote it to a file, you could have it as a bigger format, which would fill in those missing ones. Okay, so what is important, as I mentioned, in these type of models is the predictive performance. So let's do cross-validation on our SPLSDA model that we selected to see how well it predicts ones that were not used to fit the model. And we're doing this 5-fold cross-validation, so holding back 20% and using the other 80%, repeating that 5 times till all have been predicted at least once, and then we're going to repeat that entire procedure 10 times because there might be random variation in how those 20% splits are done. You want to do it more than once so that you make sure that everything's been mixed around and there's not like you didn't get like an anomalously good or bad predictive performance on one particular iteration of your cross-validation. You want to iterate cross-validation a lot of times. And here we're doing 10. Actually, sometimes you would do even more than that in practice, but that's just in the interest of time of computation. Because right, it took only a second to fit the model, but we're actually going to fit the model 50 times basically, because 5-fold cross-validation repeated 10 times. You have to run this model 50 times. So it'll still only take a few seconds, but obviously the more you do it, it can quickly blow up in the amount of time. And that's when you'd want to run it on a computing cluster SciNet, where you could run the different repetitions in parallel on multiple processors at once. And we're going to look at the error rate on our different in cross-validation. And you can see set seed is set to a random seed. We've initialized a random number generator because cross-validation splits up the data points randomly. If you do it with a different random seed, you'll get slightly different results. But we're going to minimize the influence of the seed by repeating the cross-validation 10 times. So it shouldn't be too different depending on what you set as the seed. We're going to run this. So you see it's taking a little bit longer because we're running basically the model 50 times, but it still only took a few seconds. And it tells you the error rate. Basically, there are 3 different distance metrics that it can use to predict, to assign a sample class to a class to a data point with unknown class. And you can see if we only use one component, it gets about a 40% error rate. So it's wrong about 40% of the time. As we add 2 components, the error rate goes down to about 20%. By the 3rd component, the error rate is very low. We predicted with high accuracy the class. And there's also a balanced error rate, which corrects for the fact that there's an imbalanced sample set or a sample size where we have only a few of one class and more of another. But the sample size wasn't too unbalanced, so the balance error rate is pretty close to the, it's not like we have 10 in one and 1,000 in the other. We had like 10 and 20, so it's not too imbalanced. And you can also look at the error rates for each class separately. You can see that if we only use one component, it only has 1% error for EWS. So it looks like component 1 is what's separating EWS from the other groups. But we had very high error rates for the other. If you add component 2, you drop the error rate for one of the class. And then in component 3, we've kind of able to separate all the classes and have a low error rate for all of them. And the different distance metrics have different error rates because they're slightly different in terms of the way they assign things. So then let's see, this next part, there's just various different things you can look at in terms of the output of the SPLS-DA. One of them is to look at across the different cross-validation runs, how frequently were the same, because you know sparse PLS is keeping only some of the genes in the model. So if a gene is selected across all the cross-validation repetitions most of the time, that means it's probably very important in terms of differentiating the tumor classes, because it is, regardless of how we did that random split, that gene came out on top in terms of explaining the most variations. So we can look at that. So you can see that like the stability is the how often it was selected. So we have a few that were selected about 50% of the time, and some that were hardly ever selected. So it'd be like the ones over here would be the ones you'd look at. And you can, this is just a plot, but you could also pull out the actual gene identities and their frequencies in a table as well if you wanted to look at them specifically. And there's also a function called select var, which tells you which variables were selected for each component, and like the variables being the genes. And then we can look at the ones that have the highest loadings on the components and also on their stability, how often they came up in the cross-validation. We'll run that, and you can see the genes with sort of a high absolute value on component 2 are also ones that were selected over 40% of the time. So those were like among the highest ones in terms of, so those are probably the ones that are really influential in differentiating the tumor classes. Yes, so we did cross-validation to look at the model performance of a model. We already picked out the optimal number of genes to keep, but you really should do cross-validation twice. Once to figure out what those optimal parameters should be by tuning the model, and then do another round of cross-validation on your best model to look at its prediction performance. We skipped that first step just to demonstrate things, but let's take a step back and actually do that. But this is very computationally intensive, because now we have to run the model even more than 50 times. We have to, because remember, we chose 50 variables on the first component, 40 and 30, but there's many possible combinations of numbers we could have chosen for that. So really, we'd have to run the model, do cross-validation a lot of times for many possible combinations of those parameters to use. And also, we selected 3 components of our in our PLS. Really, what if it's not 3? What if an optimal number is more than 3 or less than 3? It says in practice, k minus 1 is the optimal number, where k is the number of classes, and we had 4 classes, so we had 3 components. But that may not be the case. We really should test that instead of just assuming it to be the case. So we're doing 5-fold cross-validation. It's kind of better to do more folds than that, but to speed up things, we're just going to do 5. And then how many cross-validation repetitions we should do. And we want to, you don't want to do too many folds, because then you'll only have 1 or 2 data points you're predicting. If you do too few folds, you don't have enough data to fit the model. So 5 to 10 is kind of like the optimal way to do it. So, but actually this we can even, I included the preloaded results of this cross validation, so you don't even have to run it because it takes about 5 minutes to run. So we'll just skip the actual running of the cross validation, and we'll just load it. Now, let's take a look at the results. So this helps us, this is choosing the optimal number of components and looking at the error rate of predicting data that were not used to fit the model. You can see regardless of what distance metric we use, the error rate is extremely high for 1 component, goes down with 2. At 3, it levels off, and adding more components is not helping us anymore. So if I saw this, I would say, well, 3 is a good number. Any more is not helping our prediction accuracy anymore. Plus, it's already about as low as it can get anyway. It's down to near 0. So there's no benefit of going to higher than 3 components. So we've tuned it by number of components, but let's also do a tuning step for the number of factors to retain on each component, where last time we just picked that arbitrarily. So we could say we'll try everything going up by 10s from 20 to 100 for all 3 of the components and every possible combination, and see which one maximizes and has the best cross-validation performance. That's what you do in practice. And that takes a long time to run, again, because it's doing the model dozens of times, if not hundreds. So we're going to skip that step as well in terms of actually running it, and just load the results. And we can look at the error rate of it. And you can see yes, I think it looks like the error rate goes down as you go to 4 components. For some reason, it says 3 components are sufficient, but it looks like 4 actually makes it go down quite a bit more. So I would say 4 maybe is possibly better than 3, since it's reducing our error rate even more in cross-validation. And so this is telling us, well, according to this, the optimal number of components to keep is 3. Because you can see here is the error rate. This plot shows the error rate for the different numbers of features kept for each factor and number of components. So with 1 component, here's the error rate. 2 components, 3 components. You can see that, and 4 components is right on top of the 3 components. It's not making the error rate go down very much more when you have 4. So I'm not sure, actually, maybe this, yeah, so this is this is actually what we should look at. So it goes from 40 to 6%. Already the error rate is pretty close to 0 with 3 components. 4 doesn't improve it very much more. So that's actually the output we should be looking at for that. So based on that, I would say 4 is not adding very much, and we should stick with 3 components. And it also tells us the optimal number of factors to keep for each component as well, determined by cross-validation. Yeah, so I don't know if this is worth going through the rest of this. Well, we still have 15 minutes, so let's see what.
Could you show predict and just pick something like now we made our model, we decided how many components we want
Right.
to leave into it. We feel like we have a useful model. Now, how do we interact with this? Model is in there somewhere, but we're not looking at it.
Right.
How do I give it a line of like, here's my new patient that I've never gotten all of the data from, but here's the data I have, predict the rest of it.
Well, that's exactly what we're about to do. So why don't we go ahead and do that? Yes, we've now determined the optimal tuning parameters for our model in terms of how many components it should have and how many genes should be associated with each component, which strikes the best balance between explaining variation in the data without overfitting. So it does the best at predicting new data. We're going to basically do a little training test split on our data, we got 63 samples. We're going to take 50 of them and refit the model, our best model tuning parameters that we determined on just those 50, keeping our 13 other ones back to predict. In reality, it would be best to get an entirely other set of patients and that the model, the tuning process never even got to see. That would be even better of a test. You know, the further you can get from your data and still predict, the better the model is, right? So that's true no matter what you're doing, right? Like if you're doing if you're trying to determine what cultivar of a crop is best, it's great if you found the one that performs best in your field, but you want the one that will perform best in a new field that it wasn't tested on. So like, that's why you want a model that, okay. Don't touch anything. Yes. I accidentally bumped my link past and then it got mad at me. Yes, what you want is a model that will identify the cultivar that was best in your field. That's great, but it also should be the one that's best everywhere. But you know, that's just that's a harder test. Like if you can do that, that's where the big bucks are at, you know, so that's what you want to be able to do. We're doing the best we can here by just getting rid of pretending we don't know the answer for some of our things and seeing how well we did. But the best thing would be to get even more data, new samples of patients and predict them. But we're doing the best we can for demo purposes. So we're splitting up our data into two chunks. One has 50, one has 13. That's all this does. Now we redo our model with these optimally chosen parameters. We're basically refitting the best model that we chose using the training data only. And we also determined that this Mahalanobis distance metric was the optimal one that had the lowest error rate. So we're going to use use the model we trained on those 50 to predict the other 13 using this distance metric that also through our tuning we determined was the best. And now let's look at how that stacks up with the truth. And so you can see pretty good because and we actually are able to look for every individual in the training set, how it predicts with only one component, 1 and 2, and all 3 of them. You can see that it basically is perfect. In fact, in this case, the matrix of the truth and this is the prediction and this is the truth is perfect correspondence. All 7 of the RMS were predicted to be RMS and so forth. So I was like, wow, that's pretty good because they were not used to fit the model and it still predicted them. So really there must be something about this gene expressions that actually is different between the tumors, different enough that we can have data on the gene expression without knowing what type of tumor it is and call it. And yeah, so that's what is this is the last bit of output. It can tell you basically, right, this is calling it as 1, but it gives you the way it's being called is it's giving a score for each of the 4 and picking the one with the highest score. So that's what this shows as well. But that's yeah, that basically concludes all the content we have for this demo, and we still have about 8 minutes. So I'm happy to answer any questions about this or anything else we've talked about so far.